Bhargav Bhushan, shall we start?
Sir, వినపడుతుందా?
ఇప్పుడు ఇందాక మీద ఆఫ్ చేసేది. ఓకే,
shall
we start?
Yes, sir.
A warm welcome to respected today's speaker and dear participants for the 5th day of 1 week online international faculty development program and skill development program on emerging innovations and insights in pharmaceutical sciences, jointly organized by Jawaharlal Nehru Technological University, Kakinada, and VV Institute of Pharmaceutical Sciences, Gudlavalleru, Krishna District, Andhra Pradesh. And the eminent speaker of the today's session and my good friend is Dr. Kamal Dua, lecturer, Discipline of Pharmacy, Graduate School of Health, University of Technology, Sydney, Australia. And he will deliver a lecture in the area of pharmaceutics with a talk on targeting inflammatory diseases using nano drug delivery. Nowadays, nanotechnology based drug delivery systems aims to target the drug preload to the right place at right time at the right dose. Right dose means optimal dose. Nano drug delivery systems enhances the bioavailability of each drug, reducing deleterious side effects caused by related toxicities. In general, nanodrug delivery systems are submicron sized particles with one or more therapeutic agents that are dispersed, adsorbed, or covalently bound in encapsulated vesicles, capsules, or polymer matrices. Nanotechnology offers multiple benefits in treating chronic human diseases by site-specific and target-oriented delivery of precise medicines. One of the best example in this area is liposomes. Nanodrug delivery systems are widely used in cancer treatment and inflammatory diseases and diagnostic testing and etc. Today's speaker is having a sound knowledge in the area of the pharmaceutics, and he is also good researcher also in the area of the pharmaceutics. Now I hand over the session to Dr. P. Bhargava Rao, Head Department of Pharmaceutics and coordinator for the today's webinar series, to introduce the speaker to the participants. Over to Dr. Bhargava.
Thank you, sir. A warm good morning to the scholarly pharmacy community who are watching EFDP live on Zoom as well as YouTube. Today, it is a fifth day of 1 week online international faculty development program and skill development program on emerging innovations and insights in pharmaceutical sciences. This program is jointly organized by Jawaharlal Nehru Technological University, Kakinada, and Vallabhnendi Venkatadri Institute of Pharmaceutical Sciences, Gudlavalleru. Today's distinguished speaker is Dr. Kamal Dua. He is a lecturer in discipline of pharmacy, Graduate School of Health, University of Technology, Sydney. Today's topic is targeting inflammatory diseases using nanotechnology. So targeting is a very novel topic which has a large scope and insights to be explored. Nanotechnology is in brief, reducing the size of the particles to the nano size. Therefore, today's topic is in brief, nano-sized particles are directly targeted to treat inflammatory diseases. We can achieve many advantages, that is, we can reduce the dose and increase the bioavailability, decrease the toxicity, and extend the duration of action. With this brief introduction, now I'll go to the profile of Dr. Kamal Dua. Sir holds his dual PhD degrees in pharmaceutical sciences and immunology. His research paved the path for further studies in Professor Hansbrough's lab investigating the susceptibility of pregnant women and pediatric populations. He has successfully built collaborations with both national and international institutions, including respiratory clinicians at John Hunter Hospital and researchers across various disciplines. Sir has contributed to the field of research on influenza, infection in respiratory diseases, and has also influenced the field of drug delivery by sparkling recognition of encapsulating potential therapeutic moieties. He is an active member of research committees in GSH, including the Board of Studies, Discipline of Pharmacy, Curriculum Development, and Research. He is an active member of many national, professional, and international bodies. Sir has published around 123 publications and wrote about 6 book chapters, and 2 books are under press. Sir had won 6 grants from Department of Industry, Innovation and Science and UTS Australia and IMU Malaysia, both as chief as well as associate investigator. Sir had awarded the prestigious Mac Janet Prize. He was awarded the Young Scientist Award by the Association of Pharmacy Professionals India. He has invited as a guest speaker and several national and international conferences throughout India as well as the rest of the world. Sir has supervised 6 PhDs and 14 masters and 28 undergraduate students. Sir is also one of the panel of many editorial boards for various journals. Now, I cordially invite Dr. Kamal Duvagaru to start the session.
Can everyone see my slides? Is it visible?
Yes, sir. Is visible.
Okay, great. Just give me a minute. All right, thank you. Thank you so much for the nice introduction. And thank you so much, Dr. Rao,
Yes,
for inviting me on this event. So my today's talk is all going to be about the nanotechnology and how we can combat the inflammatory diseases using nanotechnology. So my students and my group particularly work on both biological sides as well as on the drug delivery. And I work purely on the nanoparticles and testing it for the biological interventions in inflammatory diseases. So in today's talk, I'm going to talk more about the advancements and applications in the nanotechnology in the pharmaceutical sciences. So this is the country from where I am, Australia, and that's the state where I'm based at with the New South Wales. And it is well known for two of the major icons in the world. One is the Sydney Harbour Bridge, and the other is the Opera House. I'm based at the Graduate School of Health, University of Technology, Sydney, with the discipline of pharmacy, and also associated as a conjoint lecturer with the University of Newcastle, and as a researcher with Hunter Medical Research Institute. Now, this is an institute where most of the biological studies of the formulations has been carried out, and even my second doctoral studies I have done from HMRI. Moreover, currently my PhD students are also being based at the Centenary Institute, which forms a part of both the University of Sydney as well as the University of Technology, Sydney. Now, a lot of the biological studies we do carry out at Centenary Institute with the Center of Inflammation, which is led by Professor Phil Hansbrough. And in that Center of Inflammation, I led the drug delivery node. Now, to start, actually, before I start and jump into the nanotechnology, just recently, on the 7th of May, one of our interviews is published in the Nanomed Zone. This is by the Future Science Group. And that clearly has shown in what area our group work and what exactly we are focused on. So in nutshell, my group is particularly focused on understanding the inflammatory respiratory diseases as a main focus, but it's not restricting to that. We are looking on various other diseases as well using the concept of nanotechnology. But our main intention at this stage is to look into the various respiratory diseases, including asthma, chronic obstructive pulmonary disease, pulmonary fibrosis, and lung cancer. We have identified various microRNAs and intraleukins, which are really get elevated in the conditions like asthma and COPD. And when we normally treat with the antagonists or the microRNA inhibitors, it suppresses their level. And we have shown that it has really helped in improving the pathology of the disease. So at this stage, we are trying to develop a lot of different nanoparticles of these microRNA inhibitors as well as the intraleukins. So this is the group actually with whom I work with. Professor Phil Hansbrough is the one who leads the Center for Inflammation at the Centenary Institute and the University of Technology, Sydney. And in the same center, I led the drug delivery node. Dr. Dinesh Kumar Chillappan is from IMU Malaysia, and he's one of our very close collaborator, and he worked very in parallel in developing various formulations for the respiratory diseases. Peter is from UNSW, and we have just started our new collaboration where my PhD student Minu is actually developing her sugar-based nanoparticles in his lab at UNSW. And Keshav is one of a postdoc who works with the Center of Inflammation and helping with a lot of biological studies of our formulations. Now, recently we have published in 2018, we started our first paper working with curcumin. And I think everyone knows that curcumin is a well-known natural compound, and it is a very good anti-inflammatory compound and is being used extensively in various diseases. Considering that in mind, we have also made some liposomes and with curcumin loaded, and these liposomes we have actually tested in asthmatic cell lines in vitro. So the best advantage of working in parallel with the certain laboratories is that we normally get quite a number of the human samples in terms of the human cells through biopsies. And we normally use our formulations in vitro to treat those cells to see how exactly it is controlling the inflammation. Recently, we have just published a while ago in December 2019 and another drug. So my group particularly focused quite a number on the natural compounds. We have discovered that the quercetin loaded liquid nanoparticles are also quite a good therapeutic intervention in asthma, because it has shown it suppresses quite a number of the inflammatory markers, including IL-6, IL-8, and TNF-alpha. Just now, a month ago, we published another paper with the International Journal of Polymeric Materials and Polymeric Biomaterials, in which we have shown another natural compound called celestrol. Now, in the previous paper, we have used different type of the nanoparticles, including liposomes and these plain nanoparticles. While in this paper, we have focused on the celestrol-loaded liquid crystalline nanoparticles. And these are the collaborative projects with International Medical University and the IMU and the UTS and the University of Newcastle. So you can see that some part of the formulations is being done there and the biological testing is being done with us. So in this paper, actually, most of the actually the nanoparticles which we have formed with the celestrol, they were the plain nanoparticles as well as the liquid crystalline nanoparticles. They have also suppressed the major inflammatory markers. Now, these are the projects which are run not only with Dinesh, the Thiaga and the Jitendra are also from IMU, those who have really played a crucial role in developing these formulations. So I would really like to acknowledge. This is just recently hot out of the press paper. Just a week ago, we have published a review with the Drug Delivery and Translational Research. It is a good read to know about what exactly a new kind of the nanoparticles or a vesicular drug delivery called polymersomes can be one of the best drug carrier, particularly in cancer diagnostics. We also, if you want to know more about what exactly are the patents therapeutic drug delivery in the respiratory diseases, that's another paper from our group. We work quite significantly on the biological part of the formulation. So we do testing not only restricted only to the in vitro testing, but we also had a unique mice models with the Professor Hans Blord laboratory at the Center of Inflammation. So we can test those formulations with the various mice models of the respiratory diseases as well. Now, these are the various reviews which we have published in the last 1.5 years. All are focused on the respiratory diseases. So we are trying to look not only restricted to the microRNAs or the intraleukins, we are also interested in knowing about the silencing RNAs as well as the various plants-based moieties which are being quite useful in combating the respiratory diseases. Apart from those, there are various other papers in terms of the vesicular drug delivery, which we have published in the past 2 years. And that includes not only the liquid crystalline nanoparticles or liposomes or just the plain nanoparticles. We have also worked on the green synthesis, including the copper nanoparticles as well as the silver nanoparticles. And they are being used for the different inflammatory diseases, not only the respiratory, but it can be also used for the anti-urolithiasis, as well as it has been also been used for the various other respiratory conditions. Now, this is just a bit of a background of what the area I usually work with. Now, I'm going to just share one of our study and some of the results finding. So how exactly the nanoparticles and the nanotechnology comes in the area of pharmaceutical sciences and how it works. So I believe looking at this picture, we are talking about the small things, which are tiny particles. So in this picture, in this photo, it is quite clear that these ants are trying to drink the Coke with a straw. So it means the straw need to be as small as it can be. And that's lead to the concept of the nanoparticles. And it's nothing, it's all about the size reduction. So it is quite a fundamental unit operation process, and I'm sure it has been well taught in the undergraduate studies as a whole unit operation subject where you know what exactly is a size reduction. It is basically just reducing the size of the drug particle, which can further enhance the solubility and bioavailability of the drug, ultimately reduce the toxicity of the drug, it enhances its release, and finally provides the formulation with the better opportunities for the drug. And this can be done in a various micro ranges from the powder, solution, and emulsion, and to any other dosage form you would like to target. This is a very good review which we published a while ago, and it can clearly tell, instead of I should talk a basic information about nanoparticles, I would like to show some of the data. So this review is a quite extensive, and it clearly shows what are the various challenges and what are the various prospects we have with the nanomedicine or with the nanoparticles, and what exactly need to be done in this area. So drugs, anything in the nanometer size range, it can help in improving the performance of the drug. And it had a massive advantages, and it can reduce the dose. It can also lead to the increased therapeutic efficacy. It can give us a controlled biodistribution. It can modulate the pharmacokinetics. It can decrease the toxic effects. It can give a better patient compliance because patient need not have to take the dose over and over. And the best advantage with the nanoparticles is we can have a site targeting. What exactly is the nanotechnology? It is a Latin term. It derived from a Latin term called dwarf, which means small. And it can be up to the atomic or a molecular scale. Now, it is already known in the market, and it's not only restricted to the pharmacy, it is being used in the wide different areas, including engineering and various other, electrical and other modes as well. It makes the product quite light. It makes your product strong. It has a faster efficacy. It is small and gives you a more durability. How small it can be. It is a generalized definition that it should be up to 1 to 100 nanometer in size. And it can involve in developing or modifying material or device within that size. So it should be in that nanometer size range. It is compared to the human hair, and which is about 100,000 micrometer wide. Now, this nanotechnology actually is being used, as I mentioned, in various areas, including information technology, energy and environment, advanced material and textiles, aerospace, food and agriculture, mechanical engineering, robotics, national security and defense, biotechnology, and the medicine and health. So these are the two areas, the medicine and health and the biotechnology are the one which are of the utmost overlapping with the pharmaceutical sciences. So the various kind of the biotechnological products, including the various drugs, can be delivered through the nanotechnology area or through nanoparticles. Why there is an interest in nanotechnology? At the nanoscale, as we know, we can modify the physical, chemical, and biological properties of the material. And that can fundamentally actually improve the efficacy of the compound. Nanotechnology research and development is actually not only just restricted to the material, and it's actually exploiting their properties. If you really look, this is very old data of 2005. In global investments which have been made in this area is almost 9 billion US dollars in 2005, which you can show with different countries. However, if you look at the latest stat of 2020, the US market of the nanomaterial projection is really going very well. It is up to 35,000 of the $billion are going to be invested on the area of nanotechnology, which clearly shows there is a great, huge potential and a huge demand of this area increasing in every sector, including pharmaceutical sciences. So currently, these nano applications and nanoparticle applications in medicine are geared towards the drug discovery and drug delivery, where your pharmaceutical sciences falls. Nanotechnology is a basic classification, and you can give them any name. Nowadays, people are using very fancy names to give the simple concept of a vesicular drug delivery: polymersomes, aquasomes, ethosome, nanosomes. You can call them anything. However, the main principle behind every single vesicular drug delivery system is to have a controlled release, a site-specific targeting, reducing the side effects, and providing the best patient compliance. There are various applications of nanoparticles to the drug discovery. It can be used as a fluorescent markers. It can be used in detection of proteins, probing the DNA structures, purification of the biological molecules in cell, MRI contrast enhancement, tumor destruction in cancer, and tissue engineering, and also the drug delivery or a gene delivery. Apart from that, nanotechnology is also coming excessively in various areas, including medical devices, biotechnology products, tissue engineering products, vaccine, cosmetics, and in combination of all those above. The best example I can quote here is the devices are the silver nanoparticles, which have been commonly used in the bacterial wound dressings. There are engineered calcium phosphates, which are being used in the human bone actually to give the composition and performance to the bone. It can also use for the dental restorative, cosmetics, as well as in the sunscreens. Now, looking at this, it clearly shows that there are enormous potential with the nanotechnology field. But as I mentioned, that we try to focus more on the inflammatory diseases. So this is one of the paper which we published in 2016. It's one of the old paper, but it's really, I like this paper because it had quite a basic potential which every nanoparticles cover. So in this paper, what we have done is we have developed the pro-niosomes containing lornoxicam. The basic concept behind the lornoxicam is it is a non-steroidal anti-inflammatory drug which belongs to the oxycam class. It had quite a potent analgesic and anti-inflammatory effects. It is used for the various clinical activities like rheumatoid arthritis, osteoarthritis. It can be used in the management of pre-operative and the post-operative pain, including orthopedic, gynecological, and dental surgeries. Now, the problem with these lornoxicam is it was only required oral administration. And because of its short duration of action and low elimination half-life, it was really, despite of giving with the oral route, it was not getting as effective. And there was no topical application of lornoxicam existing in the market. Moreover, as we know, these NSAIDs have always have a tendency to produce a lot of gastric irritation and peptic ulcers. So we thought, why not we just develop the topical doses form or with the pro-niosomes concept using lornoxicam. So the low solubility in the gastric acidic environment remain unabsorbed for a long period in stomach, and actually that can exacerbate the potential side effect. Now, to avoid the systemic side effects, the topical delivery of lornoxicam actually we use, it can help in bypassing the hepatic first pass effect. So the current investigation attempt was lornoxicam transdermal pro-niosome gel, and the aim was, and the hypothesis was, whether it is going to produce a sustained release of the drug or not, and it's going to help in the patient compliance and avoiding the GIP side effects. So we use various non-ionic surfactants, lutrol F6, lipoid ATH, and cholesterol. And I can show you when we have used these, how exactly they have affected the physicochemical characteristics of the drug and the pro-niosomes. So the main problem by using these, by using such non-ionic surfactant, it can actually avoid the problems what we normally have with the aqueous niosomes, such as fusion, aggregation, leakage, and the vesicular integrity. So what we have thought of, so these pro-niosomes are the dry formulation. So when you apply on the skin, it automatically gain the moisture from your skin and get changed into the niosomes and get absorbed. That was the concept we're trying to develop here. And that can provide and preserve the physical stability and the vesicular integrity. Now, this bilayer component of the phospholipid and the non-ionic surfactant in pro-niosomes can itself act as a penetration enhancer as well. We use a basic method of the co-adsorption phase separation, and these are the different compositions we have used. And how we selected these is, I can show you, and the gels were prepared by the hot and cold normal method. So Lutrol F68 and lecithin actually, they have excellent skin compatibility, were chosen as the surfactants. Cholesterol was used, which is important for the cell membrane. It's a part of a component of that, and it can also help in further enhancing the permeability and stability. So what we have done, these pro-niosomes, when they get an excess of water, they change into the niosomal dispersion. So in these pro-niosomes, when we are adding the non-ionic surfactants like Lutrol F68 and lipoid 18H along with alcohol, it changes into a lamellar liquid crystal at a craft temperature. And that's how, with the excess of water, it changes into the niosomal dispersion. And this niosomal dispersion can help us preventing various problems such as fusion, aggregation, vesicular leakage, and the stability. Now, when we have looked at the effect of the bilayer composition on the drug diffusion and entrapment, we have used the different amount of the Lutrol F68 and cholesterol. You can see that it is varying from 80 to 90 to 100, from 80 to 60 to 40. So when we have used the different composition, the lipoid 80 was kept constant, which was 80H, while only we have varied the concentration of Lutrol F68 and the cholesterol. And when we have looked at the entrapment efficiency, it clearly shown that when we are increasing from 80 to 90, there is an increase in the entrapment efficiency. However, when we are further increasing, it started reducing the entrapment efficiency, and that makes us to select the 90 meg of the surfactant is the optimum for the pro-niosomal gel. So as we are initially increasing the concentration of the surfactant, it increases the number of niosomes formed. And that actually can further increase the volume of the hydrophobic domain and increase the entrapment efficiency. However, when we are further increasing the concentration, it might reduce the entrapment efficiency due to the mixed mesial formation with the niosomal vesicles, and that is further increasing the entrapment efficiency. After that, what we have thought of looking is the entrapment efficiency of the other component, which is the cholesterol. So cholesterol are being used in three different 80, 60, and 40. And you can see clearly that when it's being used from 80 to 60, the percentage entrapment efficiency increases. While when it's been reduced, it started further reducing the entrapment efficiency. And that entrapment efficiency is actually due to the molecular cavities of the surfactant bilayer, and that was abolishing the gel to the sol transition. And that is forming a leaky vesicles, due to which there is an increase in rigidity and reduce in the permeability of the drug. And finally, it's leading to the improvement in the entrapment efficiency. So with this, we selected the 60 mg of the cholesterol, and that makes us the standardization formulation along with the cholesterol. And these are the same components why we have used that, and it's further helping partitioning of the drug in the bilayer and increases the entrapment of the drug to the niosomal vehicles. So when we have looked at the in vitro diffusion, it has clearly shown us that there is a sustained release we can see. And this sustained release actually is sufficiently longer period of time to avoid frequent applications and provide the better patient comfort. When we looked at the particle size and the polydispersivity index, it was quite optimized, and they were well settled what is being needed for the nanoparticles, including the appearance, size, zeta potential, polydispersivity index, and the entrapment efficacy. And when it was being checked through the ex vivo skin permeation study, it was really shown that how exactly it is showing a sustained release as compared to the plain drug when it was being loaded in the gel. It has also shown quite a spherical structures with the pro-niosomes when being checked through the SEM. And also there was no compatibility was found with the DSC, as well as it can clearly show that the crystalline nature of the drug was being changing to the amorphous as they are getting into the pro-niosomal form. So with this set of a study, it was quite clear that pro-niosomes can be a potential for the transdermal drug delivery of lornoxicam over the conventional gel formulations. Now, if I go back to one of my previous slides, and where I have shown you the various kind of the drug delivery we've been working on, so you can clearly see that not only restricted to the transdermal drug delivery, we are being also working on particularly in cancer. Recently, our group had also encapsulated a couple of new drugs like rutin, naragenine, and berberine. These are the drugs which are based from the phytochemical background, and when they have been encapsulated into the solid lipid nanoparticles, and we have tested for the anti-cancer, particularly in the lung cancer effects using the various in vitro techniques like apoptosis assay and the migration and proliferation. Particularly, we have found that they all been driven through MMP9. It is one of the metallomatrix protein which is being controlling the inflammation. Apart from that, also, we are also interested in looking at various kind of the oxidative stress genes. So oxidative stress is one of the major biological phenomenon which is happening in the cells. And this oxidative stress is actually leading to the inflammation in these drugs, whether it is asthma, whether it is chronic obstructive pulmonary disease, which you call emphysema, which is highly prevalent in India due to cigarette smoking, or the pulmonary fibrosis and lung cancer. So when we have seen those kind of the herbal potential drugs are being encapsulated into these drugs, they have really shown quite a high degree of the potential in treating those inflammation in those diseases. I'm going to share one of the small presentation of quercetin, what we have been already published, and I have shown you that how exactly the quercetin liquid crystalline nanoparticles have been shown in reducing the inflammation of those markers in the respiratory disease. So can you see my new slide, a new set of slide? So what we have done here is we have prepared the quercetin loaded we have prepared the quercetin loaded liquid crystalline nanoparticles, and we have checked its therapeutic intervention in asthma. So as we know, asthma is one of the chronic airway disease, and it is particularly characterized by certain features such as airway inflammation, airflow limitation, hyperactivity, and airway remodeling. The basic treatment strategies currently existing includes inhaled and systemic corticosteroids and the beta adrenergic agonist. So, but when we looked at these treatments, the people normally, or the patients normally, they get the steroid resistance, which is the steroid insensitivity. Once they start getting the steroid insensitivity, it actually causes a lot of problems, including starting showing the side effects to the patient. It means that treatment is no longer effective. So what we have done here is we thought of quercetin using as a drug, which is one of the dietary flavonoid, and it found in fruits, vegetables, including onion, apple, grapes, wines, and nuts. It has a quite well known for the various therapeutic uses, including anti-inflammatory, antimicrobial, antioxidant, anti-allergic, and anti-cancer. It has an extensively one limitation. It has really poor aqueous solubility, and its direct consumption actually does not provide the therapeutic effect which is being needed. When we have looked at the Therapeutic Good Administration website in Australia, it's still listed under the complementary medicines. It means people can still, in Australia particularly, you can use that. And you would be really surprised that in Australia alone, the market for quercetin formulations was unbelievable. these are just few of the products which have been existing in the market with quercetin alone as well as in the combination. So we thought, why not we check the potential in asthma? And it was well known. When we looked at the literature, it was clearly shown that it has been a well known in anti-asthmatic action as well. How it's normally show its action, it has been also been already well literatured. Through the mast cell degranulation, it also reduces the eosinophil recruitment as well as neutrophils, which are the eosinophils is one of the major cell which plays a crucial role in managing the in the pathophysiology of asthma. And it also reduces the airway hyperresponsiveness, which is one of the key characteristics of the respiratory diseases. And finally, it also reduces a lot of the pro-inflammatory cytokines. So the application of the quercetin in clinical practice was still limited because of these two reasons. And that made us to think, why not we make the liquid crystalline nanoparticles? So we used a similar sort of a procedure to develop these liquid crystalline nanoparticles. And these liquid crystalline nanoparticles have given us various advantages. First of all, they are bioadhesive. This is what we need so that if there's a lot of mucus, when a patient gets asthma, there's the goblet cells start secreting a lot of mucus. And because of that, it can easily, these nanoparticles can easily stick on the epithelial cells. And once they get stick on that, they will continuously start releasing the drug through the prolonged duration of the time, and which can going to be effective for the patient, providing a sustained release behavior. These kind of the liquid crystalline nanoparticles are having greater hydrophobic and drug loading capacity and a better physical chemical stability. So we use the similar sort of the composition and we develop these using a standard method. And these are characterized for the usual, which everyone do around the globe for the physicochemical characterization, zeta potential, particle size, polydispersivity index, incompatibility studies, and the in vitro release. And then the most important thing in this was the anti-inflammatory potential. So we have looked the anti-inflammatory potential using the primary bronchiothelial epithelial cells. Now, so it has clearly shown that there is a significant increase in the particle size due to the encapsulation of the quercetin as compared to the blank nanoparticles. PDI value was less than 0.3, and that was showing the narrow size distribution. And also, the higher PDI values of the quercetin liquid crystalline nanoparticles are attributed to the coexistence of the cubic liquid crystalline nanoparticles with the other type of vesicles. And the positive zeta potential was actually reducing the chances of aggregation. Now, when we have looked at the morphology, they were quite a bit of a spherical in size, and the size of the particles was less than 300 nanometer. And we have looked at the in vitro release studies, and it was quite showing the sustained release over the period of time. These are the primary bronchioepithelial cells, which we normally derive from the patients and we culture in the lab. Now, this is the air interface cells which we have grown, and these cells, we can not only in our group, we are only restricting from asthma, studying asthma, but we also look for COPD, which is the chronic obstructive pulmonary disease. So these cells are normally being grown, and once they are grown, they are being treated with the various formulations. Once you treat these with the various formulations, then you can actually take out the supernatant, and these supernatant, you can test it for the various type of the pro-inflammatory markers. They can be extended to the major pro-inflammatory cytokines. It depends on the nature of your nature of your disease, which kind of the disease you are understanding, whether it's a COPD, whether it is an asthma, or you targeting the lung cancer or pulmonary fibrosis. So in this particular case, we have looked more on the asthma. So the major cytokines we have looked actually is the IL-6, IL-8, and IL-1 beta. It has clearly shown that there was an increase when the patient is having asthma. However, when it was being treated by the liquid crystalline nanoparticles of the quercetin-loaded liquid crystalline nanoparticle, it started reducing the inflammation. So the level of IL-6, IL-8, and IL-1 beta start suppressing. Not only restricted to this, there are certain data which is not been included in this in my slides are from the anti-cancer potential. We have really seen tremendous effects when we have done the apoptosis and the proliferation and migration assays, and it has clearly shown that it is particularly showing a significant reduction in the inflammation in the cancer. Now, it is only the in vitro inflammatory activity, and that we are believing that it might be through the NF-kappa B signaling pathway, which we still need to identify through which pathways they are working. And similar sort of the studies is being done on the various other papers which I have shown you before. Now, apart from that, we also have the various mice models. So we run the various different mice models in asthma and COPD using the OVA, which is one of the commonly used model, OVA albumin-induced asthma. So in which we have been looking at the different parameters like the increase in the cytokine levels, such as IL-13, IL-6, IL-8, IL-1 beta, and also the production of the mucus using PAS staining, as well as the airway hyperresponsiveness, as well as the eosinophil staining. So when we treat these to the mice, it has also shown all these features have significantly reduced. Not only restricted to the albumin model, we have also been looking on the house dust mite-induced asthma mice model, which is more clinically relevant as compared to the albumin, which is a basic model being used. So on top of that, in the COPD, in Professor Phil Hansborough laboratory with the Center of Inflammation, there is a unique smoking model in which when we smoke the mice for the 8 weeks, through the nose-only channel, it has clearly shown that the mice develop all the important hallmark features of COPD, including reduction in the, there is an alveolar destruction, quite a significant, and when there's an alveolar destruction, it clearly shows there is the exchange of the gases cannot take place. So person having a start getting difficulty in breathing. It has also shown there's an excess in mucus. All the important hallmark feature it has been used. So those kind of the models we are trying to use for these formulation to test further. Now, in conclusion for this, it has clearly shown us that the liquid crystalline nanoparticles have quite a significant anti-inflammatory potential in treating the asthma. Similarly, when we have looked on the other drugs such as curcumin, or we have also looked at or celestrol, they all have clearly shown that it has reducing the anti-inflammatory activity and the potential. So these are the different activities what we are working particularly at present in our group. We are welcoming quite the number of collaborations around the globe. The major collaborations we had for the formulations is with the International Medical University, Malaysia, and here in Australia, in Pala, we work with University of New South Wales, University of Tasmania, and University of Newcastle, and Queensland University of Technology with Trudy Collett. We also have an extensive collaboration with the Ulster University in UK, where we work primarily on the lung cancer side of the niosomes and the vesicular drug delivery. At this point of time, we are welcoming more the collaborations around the globe to see if we can work together and we can find there would be any further potential or chances. This is another paper of us which have shown that solid lipid nanoparticles of the mometasone fureate. Now, this one, we have also exactly, I'm showing you because the way we have done with the pro-niosomes with the lornoxicam, here also we have done the solid lipid nanoparticles using the mometasone furet. And that was just to overcome the conventional formulations problem that can reduce the side effects and all those things and can help a better increase skin deposition. So that ends with my presentation. And so I have just shown you a couple of my results. Some were from the published paper, while we do have certain data, which when I had talked yesterday to Dr. Lakshman Rao, he mentioned not to include on the slides because it's going to be live on the YouTube. Now, there are certain data what we have with the anti-cancer and the anti-inflammatory potential of the other drugs, as I mentioned, like butane, narogenine, berberine, all these drugs. So it's still in the pipeline and we have not yet published. So that's why I have not included that. Now, I would like to acknowledge the University of Newcastle, particularly Professor Phil Hansborough from the Center of Inflammation and the Alan, who had really helped us doing quite a number of the in vitro activities with these cells. I would also like to thank the University of Technology, Sydney, with the Graduate School of Health, Discipline of Pharmacy, where I'm based at. Professor Charlie Benrimoj was the previous head of the school, and now Professor Joanne Gray. I would like to acknowledge her as well. Professor Kylie William is the head of the Discipline for Pharmacy, and Professor Mary is one of my collaborators who works quite tremendously on the lung cancer axis. So Mary's area is primarily more on the microvesicles, looking how they are being involved in the cancer progression and how it can be controlled. And we are on a verge of looking our formulations in targeting those microvesicles as well. So at this point of time, my students, my PhD students, they work in both the areas. They work in not only just restricted to the drug delivery where they are making the formulations, but they are also been working in the biological side in understanding the pathophysiology of the respiratory diseases, highlighting in the lung cancer and the oxidative stress. And I would also like to finally thank VV Institute of the Pharmaceutical Sciences, particularly to Dr. Lakshman Rao. Now, this is one of the beautiful picture of the Newcastle. I haven't got a photo of Sydney, and it's just on really on our Hunter River. You can see that. And it's such a center view of the city. If you get a chance visiting Australia anytime, either any part of the Australia, and if you are visiting Sydney, Newcastle is just 4 or 3.5 hours towards from Sydney. Please do visit Newcastle as well. I'm not promoting or advertising for the tourism, but it is one of the beautiful lost island in the world you can explore. While if you are coming to Sydney, please do let me know. I'm more than happy to take you around and to be your host. With this, I would like to end my presentation. And I would like to also acknowledge all my collaborators whose data I have shown you around from IMU Malaysia, from University of Newcastle, as well as from the Ulster University. It's quite open for the question and answer sessions. If you got any, I'm more than happy to answer any questions if you got for me. With this, thank you so much.
Thank you very much, sir. Dr. Kamal Dua. Is there any questions from anyone? Huh? Sir, can you hear me, Kamal Dua, sir?
Yes, I can hear you. Yes.
Yeah. Thank you very much, sir, for your valuable session. And you have done a lot of research on this. And we expect the same in future also.
Thank you.
Sir, at present, you're working on which molecules, sir? Drug molecules?
So at this point of time, my group is particularly focused on two things. It is not only restricted to the natural moieties. Natural moiety is our additional area on which we're working, but that is not our prime focus. Our prime focus area is targeting microRNAs and the interleukins in respiratory diseases using the antagonists or the inhibitors. So we are primarily have found that microRNA 21, microRNA 125A and B, they are really being elevated in the diseases like asthma and COPD. Particularly, I have worked extensively on the influenza virus, and now it's a time of coronavirus. I think that's coronavirus actually had led to this webinar, and that's why I'm there with you all. So it's coronavirus is also a kind of a virus, but it has a different actually the way it works from the influenza virus. While influenza virus is a common cold, which everyone easily can get when it comes to the cold weather. So what we have shown is when a patient with asthma and COPD, they get influenza along, their pathophysiology really changed and the patient had an increased susceptibility to the influenza infection when they have asthma and COPD. So in those cases, these microRNAs, particularly microRNA 21 and 125A and B, it really increased the levels. So what we have done, we have given the inhibitors and it has suppressed the levels. So when they have suppressed the level, it has improved the pathophysiology of the drug. It has reduced the mucus level. It has reduced the inflammation. It has reduced the eosinophil count, and as well as with the COPD, all the hallmark features. So at this point of time, we are trying to develop these nanoparticles containing those inhibitors and targeting those respiratory diseases. And in that, we are trying to do looking at both the in vitro studies as well as the in vivo using the mice models. But the in vitro, what we do is all on the human samples or the human cells, what we normally get from the human patients from the hospital. Because the Hunter Medical Research Institute in Newcastle, it is just next to the John Hunter Hospital, and similarly Centenary Institute is next to the Royal Prince Alfred Hospital. So Phil and actually everyone got quite a number of collaborations where you have these with the clinicians, where you get those samples easily and you grow those cells and then you can treat with your formulation to check the things. Now, I can see there is one question which came through the from Nataraj KS. It's talking about stability issue of nanoparticles. I think it is really quite interesting actually, because the stability is one of the important physicochemical parameter which need to be addressed. And it's not easy that everyone who make the nanoparticles will get the right stability. While it took us the study which I have shown you of the pro-niosomes, it is in collaboration with one of my collaborators from Pune, and it took us almost around 2 years to just establish the stability of those nanoparticles. So I completely agree with you. However, I think there are very limited formulations which have really gone to the market, which are having the encapsulated form, while there are a lot of cosmetics which are coming in the market have the liposomal formulations. So I do agree with you. The stability is one of the concern and it as a physicochemical properties, but it depends on the composition and the different variables which you use in your formulations. If you play around with those, as I have shown you in my pro-niosomes, that the different concentration of the non-ionic surfactants, including the different concentration of the cholesterol, when we have played around, we did manage to get a stable nanoparticles. I hope this answers your question. So what's the difference between the influence on the particle size and the zeta potential? A very good question. Very good question. So the particle size, actually, we normally also do it with the polydispersivity. So polydispersivity index is a measure of the heterogeneity of the sample based on size. And these, so if we are talking about the stability, it means we're talking about the aggregation, agglomeration. So this polydispersivity index actually govern that. So polydispersivity index can occur due to size distribution in sample. And this polydispersivity index can obtain from the instrument which we normally use, like dynamic light scattering and all. If we talk about particularly about the zeta potential, it is the electrical potential, as we know, and it is an interface which separates the mobile fluid from fluid that remain attached to the surface. So it means it is more or less, it is a scientific term, what we are using for the electrokinetic potential in the colloid dispersion. So the particle size actually plays a quite a significant role in controlling the zeta potential. So you don't know if you are increasing the size of the particle, that might be altering the size or the charges of your particle around. It means ultimately it is affecting your zeta potential. So you have to actually just play around between the particle size and the zeta potential and PDI to see where it best fits. What do you mean by the best fit is where you have no sedimentation of the particles, they're not getting too bulky, they are not clumping together, and that's how it started showing the stability. And at the end of the day, what you all need is a sustained release of the drug. So that particular sustained fashion of the release of the drug also depends a lot on your particle size, because this particle size of your nano size is ultimately altering your bioavailability of the drug. So actually it is just a bit of a play around the various variables. I hope this answers your question. So at the end, I would really like to welcome the collaborations. If you want to work with us, if you think of any ideas in which you think that we can work together in terms of the area of drug delivery. And I'm not restricting only to the drug delivery, as I mentioned, because I work purely on both the ends, the biological sciences and the formulation sciences, and the overlap and the interface of both. So if you think of any work which you think we can perform together in collaboration, please do drop a line to me and contact me. And feel free if you got any other questions in regard to the work I have presented. However, I would like to again emphasize that there are various other studies which have enormous biological data which I have not shown today because it's still not yet published. And we are on the verge of publishing. And so because of that, there are certain slides which I have to cut off yesterday after talking to Dr. Rao. So any other questions before our session?
Thank you, sir. Thank you for your session. And now the feedback link is in the chat box. Sir, next I will hand over the session to Dr. A. Lakshman Rao Garu, today convener.
Thank you, Dr. Bhalgabhushan Garu. And first of all, let me thank today's speaker, Dr. Kamal Dowaji, for your excellent presentation on the nano drug delivery, especially applicable to the inflammatory diseases. Really, you got the excellent feedback in both Zoom platform as well as YouTube link also. Thank you very much, sir, for your novel topic in the area of pharmaceutics for the benefit of the student as well as a teaching fraternity. On behalf of VV Institute of Pharmaceutical Sciences, I would like to take this opportunity to express my sincere gratitude and thanks to our today's speaker, Dr. Kamal Dua, for his plenary lecture. And I especially thank all the participants for their interest towards our faculty development program as well as skill development program. And let me thank my colleague, Dr. P. Bhargava, Head Department of Pharmaceutics and coordinator for today's webinar series for his excellent coordination. And we value to all of you and for your continued support for our programs. And tomorrow, day 6 is the last day for our faculty development program as well as skill development program. And tomorrow, the day 6 that will start at 2 p.m. 2 p.m. The subject area for tomorrow's session is pharmaceutical analysis, and the speaker for tomorrow's session is Mr. Gnanananda Varma Buddharaju, Global Leading Head, Pfizer, United Kingdom. He delivers a lecture on pharmaceutical labeling and overview. Once again, thank you, Anandal. Have a nice day. Thank you very much,
Dr.
sir.
Rao,
Tell me,
I
sir.
think before we end the session, I would just like to talk about one of the course we had at our university. I will
Definitely,
take
sir.
minutes if that's okay.
Definitely, sir. Definitely.
Okay, so can everyone see these slides, please?
Yes.
Can,
Yes.
okay,
It
so
is okay, sir.
great. So we have one of a unique course at the Graduate School of Health Pharmacy, which was not my idea to present today, but I thought I'll take this opportunity because there are a lot of students as well.
Yes, it is a good idea. Yes, sir.
So we do have a unique course in good manufacturing practice. And this is the only course in the whole Asia Pacific region which UTS have. There is none other university in the whole Asia Pacific region which had any course related to the good manufacturing practice. I am the course coordinator for the program, and I also teach in pharmacy. These slides are from one of the information sessions. So please excuse because Jacqueline normally delivered that session with me. That's why her name is there. I'll just quickly take you through on this session. So this good manufacturing practice program, actually we have developed in partnership with one of the industry collaborator. It means all the teaching on this program is done through the industry people. And this is C Pharma. C Pharma is one of a big consultancy company which is based in Melbourne. So we developed this program in collaboration with them. And this program actually is really quite exclusive in itself because it had a lot of opportunities. And we do get quite a number of international students. We do have international students from India, Nepal, Sri Lanka, Pakistan, US, South America, and Australia and New Zealand. So we got 3 different type of courses I can show you in a minute. And all these courses, among the 3 courses, 2 of the courses we have both online and on campus. Now, this is just group of students, the first group of the students when they were here. So you can see that that's quite a bunch from around the globe we have on our course. Now, this course is actually forms a part of the discipline of pharmacy because it comes within the umbrella of discipline of pharmacy. Professor Kylie Williams is the head and I'm the course coordinator, and rest of the lecturers are actually from industry, and there are various associates which come from industry to teach on this program. Now, in this program, there are 3 programs: Master of Good Manufacturing Practice, Graduate Diploma in Good Manufacturing Practice, and Graduate Certificate in Good Manufacturing Practice. The Graduate Certificate is a 4-subject program. Each subject is 6 credit point. That's why it is 24 credit. Graduate Diploma is a 8 subjects. I'll show you the subjects in a minute. And that's how it is 48 credits. While the master's program, which is a postgraduate degree, and it is the only degree in a whole Asia Pacific region, which UTS offers, of 2 years program. And the best part for the international student of having this degree is, once you come to Australia to do that 2-year degree, you automatically get 2 years work right visa. So it total, you are actually spending a 4 years time in Australia. So far, there are a lot of international students from India and other parts have come through, and we have well absorbed into various jobs around Australia. The best part is we have 2 intakes. We have autumn and spring. And now this point of time, the next intake starting is July, and the applications are still open. However, if any of the Bachelor of Pharmacy final year students, those who are finishing, or a master students, we do have students from engineering background on this. We do have quite a number of Master of Pharmacy students, those who have come to do this course with us. So it is really wide open, this course. The last 2 courses are both online and on campus. While for the online program, we have quite a number of people, those who have registered are from industry around Australia and New Zealand. Now, you can see that there are 16 subjects, and all these subjects are very industry specific. There is not even a single subject which is just a basic what you learn in your Bachelor of Pharmacy or a Master of Pharmacy. You have been given the well in-depth knowledge by the industrial experts, those who teach these subjects. When you do the master program, the industrial research project is a 1, which normally we try to get a project with an industry in Australia, around Sydney area. So you have an opportunity to go there to do your project. And what is actually happening when a student go to do a project with a company, if they really are very good and they have really shown their enthusiasm while doing their project, the company absorb them for the job. Because when you finish this course, you have 2 years work right visa. It's a postgraduate sector visa. You can bring your immediate family as well to Australia. In all the 3 different programs, the master program, the graduate diploma, and the graduate certificate, the first subject, which is international GMP and quality assurance, is a compulsory subject with all the 3 programs. While if you are doing the diploma, you can have the first 8 subjects. But if you are doing the certificate, international GMP and quality assurance is compulsory, and you can select any 3 subjects. You can do it from India if you want to do it online, but if you are interested to come to Australia, you can enroll into the on-campus program as well. This is just for the online. So I don't think any of the international students are normally interested in online program. They usually try to come to Australia to do the on-campus programs. It has enormous potential. Australia had a very growing market in the clinical trials. And they are looking for the people, those who really have a skill in GMP can apply to the clinical trials. The regulatory affairs is another area. And I'm sure everyone is well aware of the rest of the areas in industry. These are some of the stories of our students. Prashant is from India actually, and he's from Gujarat. He came to do the Master of Pharmacy with us, and he finished almost 6 months ago. And he did his project with a company called Pharmaco Biotechnologies in Sydney. And he's absorbed as a quality assurance person in that company. He's working with that company now. Shoki, which is in the middle, she is a medical doctor. She was having an MBBS degree. While she was really passionate about the regulatory affairs. So when she came to do the master's program with us, she started working with the Smith and Nephew. And she did her project with Smith and Nephew, the master's project, and she was absorbed there. But she had recently changed to another company now, because after gaining experience, when you started rolling your experience in Australia from one company, you started getting opportunities to get job opportunities from the other companies around. Anjali Patel, she's from Gujarat. She was having a Bachelor of Pharmacy. She came to do Masters, and she's working with one of the leading company in Australia, where she done her project with Innova Pharmaceuticals. So there are so many stories. There are a couple of those. But you can read these stories, the extra stories, if you want to read, or you want to read these in detail. You can just go to the Graduate School of Health UTS webpage. And if you go to the student success stories, you'll see those stories. Now, we do have certain prerequisites when you apply for this program. If your undergraduate or a postgraduate degree is in any of these areas, you are eligible to apply. If you don't have an undergraduate or a postgraduate degree in this area, but if you are having an industrial experience, any industry, food industry, meat industry, dairy industry, where GMP principles been applied, and you can show that through your CV to us, we still consider your application. And we have various students from industry. I'll give you an example. We have a student who actually came to Australia as a permanent resident 10 to 12 years ago. He was only having a diploma in electrical engineering, and he was working with the cyclotron facility in a hospital. So the cyclotron facility is the one where you produce radiopharmaceutical to test the nanoparticles. Oh, sorry, to test the cancer. Sorry, I'm still in nanoparticles, sorry. And because he was looking after the machine, the electrical parts and everything, he started looking at a bit of a quality control aspects. That really triggered him to do something in this area. So he approached us. Now he was having enough experience before the working with that cyclotron. He was with GE Healthcare. So looking at his CV, it was having almost 10 years of experience working in different healthcare industries. We offered him master's program. He's finishing in the coming semester. So you can imagine now, if a person who don't have even a bachelor's or any master, he's doing his master degree, a postgraduate degree, finishing with us. So considering that, if you know anyone who got an experience in industry and want to apply for such a course, which can give an opportunity to come to Australia to study, this is the course for you. Now, you have to apply online. For the coming semester, I'll show you the deadlines, and for the autumn next year as well. It is straight online process. You do have to meet the English requirements. It means you have to sit for IELTS or a TOEFL or whatever you say, PTE. You can check the scores from our website. You do have to meet. There are certain scholarships for this program. The first scholarship is a recently announced scholarship, which gives 25% of the tuition fees of that program. You have to actually tick that while applying your application. The second scholarship is an automatic, it will be considered when you submit your application, but it depends on your academic merit. If you're falling in that academic merit, you might get the $5,000 towards your first tuition fees. The third scholarship is a one, which gives you 100% full tuition fees waive off. This scholarship is highly, highly, highly competitive. This is a scholarship which you have to compete with the various applications which are coming from the different schools in the university. If you are really thinking your percentage in India in pharmacy, it is above 90 or 95, then you might think your chances are pretty good getting this scholarship. Now I'm going to talk about an excellent scholarship, which is the last scholarship called Australian Award Scholarship. This is a scholarship which India also come in that category to apply. This is a government scholarship. This is nothing has to do with the university. We have 3 students at this point of time on this scholarship with us. 2 from Indonesia and 1 from South Africa. So any countries which fall in the Indo-Pacific region, they are eligible to apply for this scholarship. This scholarship will not only give you the tuition fees, this scholarship will give you the living allowances as well to stay in the country for the whole duration of the time you are pursuing the course with us. This scholarship will also give you another traveling funds, plus also certain funds for your professional development. So have a look on the website through Google and see if you are eligible to apply for this scholarship. This is called Australian Award Scholarship. So these are a couple of the students, they were from our info session. Now for the international students, the application closing date is 15th of June, 2020. Whether you are in India or you are in Australia, the closing date is 15th of June. However, because I understand the borders are still closed and because of the coronavirus, no one can travel, you can still apply for the autumn, which is the next year. So any of the Bachelor of Pharmacy final year students, those who are completing, or Master of Pharmacy in any discipline, relevant discipline, particularly from pharmaceutics or the quality assurance, they are quite, actually this course is really been aligned to their previous thing. If you got any queries about the course, you can drop an email to gsh.future@uts.edu.au. If you can write to this email, they would be able to answer you. It's called gsh.future@uts.edu.au. If you got any questions regarding this program, I am more than happy to answer regarding the program because I'm the course coordinator for this program. So you can ask me anything if you have any questions about this, anything. you got any questions for me, please feel free to ask. If you want to ask anything about this program. Yes, I think there is one question I'm sure which is triggering everyone's mind about the fees. So what would be the fee structure for master in GMP or graduate diploma in GMP or graduate certificate in GMP? So I can give you the best advice. You can just go to the UTS website, University of Technology Sydney website, and you can just try to find the fee calculator. In that fee calculator, when you are going to put the course, you will get the fee structure. Now remember, when you pay here semester wise. You don't pay the whole course fee. It is always semester. And you can also pay as per the number of the subjects you are taking. It's nothing like that you have to pay for the whole course. So most of the international students which they come here, the best part of the program is the classes are only 2 days on campus. Only 2 days on campus. So it means the rest of the 5 days, students still, because they are on the postgraduate sector visa, they get work rights. So I'm not sure what exactly is at this point of time is the duration of the work rights they are allowed to work, but they start working in that area. If you get a job in an industry, it is really good because you are doing this degree and side by you are actually pursuing job. However, in case you are working on the other side, students normally easily make their living allowances. So it means all their rent and the living allowances they usually can earn while working those 4 days or 5 days, because classes are only 2 days on campus. And after you finish the course, you get 2 years work right visa. And if you're lucky enough, you'll find a job. So this is another beauty of the course. But please, if you want to check the fee structure, just go to the fee calculator on the UTS website. Or alternatively, if you can write to gsh.future@uts.edu.au, you would be able to actually get the answer. Yeah, so I got a question on a fee structure. So this is what I'm saying. If you just go on the fee calculator on the UTS website, that will give you the answer. What's the fee structure? Depending on what course you are looking for. Are you looking for a master's program or a graduate diploma or a graduate certificate? Also, feel free to write to the email gsh.future@uts.edu.au, and someone from the professional staff would answer your query within a day time. Any other question? If you come to Australia, it will really experience your multicultural experience while studying, because we got students from all around the globe on this program. Please feel free to ask if you got any question. And thank you so much, Dr. Rao, for allowing me to present the course, about the course.
Kamal.
I really appreciate it. Thank you so much.
Thank you very much, Kamal.
And thank you very much, everyone, for attending my talk. And it's a pleasure having you all with us. And feel free, you can get my email ID if you just Google it. My name, you will find my email ID. Please feel free to drop a line if you are interested in knowing more about anything which I have presented today, or want to discuss anything further to work together or in terms of establishing any collaboration. We are always welcoming that. So thank you so much again, Dr. Rao. Thank you so much to VV Institute of Pharmaceutical Sciences. And thank you each and every one of the participants, those who are there for this webinar. Thank you so much.
Thank you, sir. Thank you very much, sir. Thank you for all the participants, and you can fill the feedback, and the link is given both in YouTube as well as Zoom app. Thank you, everyone. Thank you, sir.
Thank you. Can I leave now?
Yes, sir. Yes, sir. You can. Yeah, yeah.
Thank you. Thanks. Bye. Thanks. Thank you.